My name is Aaleen Zhera, and this is my thesis project on the metabolomic effects of mild heat stress on rainbow trout blood plasma. My project is an extension of Sushmita Srinivasan's thesis project in 2020, however, due to COVID, it was cut short. To start off, what is metabolomics? Metabolomics is a large-scale study of chemical processes involving metabolites such as amino acids, fatty acids, nucleic acids in the organism's cells, tissues, and organs, while lipidomics is a subset of metabolomics focusing on analyzing lipids and fats. The molecules are separated on a liquid chromatogram, ionized into gas, and targeted using a mass spectrometer for mass analysis. As we continue to observe, climate change raises global ocean temperatures. It is predicted by the end of the 21st century, global surface temperatures will be increased by 1.5 to 2 degrees Celsius. As you can see on the figure on the right, ocean temperatures have been on an incline in a linear fashion. This is important to note because rainbow trout are ectotherms and are sensitive to these temperature fluctuations, and the effects on the aquatic ecosystems and their species will continue to intensify. This will lead to the stress response. It is known that once animals are exposed to stress, the hormones released can be induced at various levels. It has been recorded that heat stress can cause physiological, metabolic, reproductive, and osmoregulatory fluctuations in cold water species. As you can see on the figure on the right, pink salmon has primary, secondary, and tertiary responses to external stressors. We aim to build upon the present knowledge by analyzing the blood plasma metabolome. So understanding the fluctuations in heat stress responses in rainbow trout is vital to their health, performance, and impact on our global aquaculture. To start my project, I identified 3 commonly known stress molecules, which were cortisol, epinephrine, and norepinephrine. These have been seen in acute and chronic stress responses and circulate in the blood. Thus, these are suitable molecules of interest. We use deuterated standards of these 3 molecules to analyze the heat stress, as well as a non-targeted approach for measuring significant amino acid and lipid concentrations. My main study question is, does mild heat stress affect the rainbow trout metabolome, and if so, to what extent? I aim to study these adaptations of rainbow trout reflective of our warming ocean temperatures. We hope to reach this goal by analyzing 3 study objectives, which is identifying significantly varying levels of cortisol, epinephrine, and norepinephrine, as well as amino acids in the aqueous phase and lipids in the organic phase. I hypothesize these 3 stress hormones will be increased in circulation as well as overall lipid and amino acid levels in the plasma. We will now move on to the methods utilized. Sushmita set the environmental conditions and collected the blood samples. 40 rainbow trout were housed at 50.5 degrees Celsius for 1 week, and half the fish were housed at 17.5 degrees for another 7 days. The feeding, water filtration, and other controls remained constant. After the 2 weeks were done, non-lethal blood sampling was utilized to extract blood from the trout's caudal vein. The blood plasma was removed from the blood sample and frozen in the -80 degrees Celsius freezer until sample extraction occurred in 2021. The plasma samples were prepared using a modified Blyde-Dyer method and centrifuged to create 3 layers of separation. The aqueous and organic layers were extracted and the protein disc was discarded. We added cortisol D4, epinephrine D6, and norepinephrine D6 standards in both aqueous and organic phases, the amino acid standard in the aqueous tube, and the lipid standard in the organic tube. These were dried down, placed onto HPLC vials, and run on the LCMS machine for targeting. Once the run was completed, the metabolites were identified using Mass Hunter softwares and statistically analyzed using MetaboAnalyst. I will now break down the results of the data analysis. Starting with the aqueous metabolites, 17 were identified as significantly changed in abundance, 7 increased and 10 decreased due to heat stress. The metabolites are listed in increasing fold change values, with the highest ones at the tip of the arrows. To start, this principal component analysis, or PCA plot, indicates 2 groups were indeed identified based on the similarities within them. The shaded region represents a 95% confidence interval, blue being the control and orange being the heat stress group. This PLSDA plot shows the cross-validation results, as all components have a high accuracy and correlation near the coefficient of determination value of 1. This volcano plot further displays the most significantly changing metabolites, with asparagine being the most increase in abundance and threonine being the most decrease in abundance, as circled in green. This enriched pathway analysis shows us the most enriched pathways based on the hits observed for each amino acid. As you can see, threonine 2-oxybutanate degradation was the most enriched pathway found in the aqueous metabolites. Moving on to the lipid metabolites, 36 were significantly identified, with 27 lipids increased in abundance and 9 decreased. To further visualize the significant lipid profile, the figure on the right shows 5 categories of phospholipids and their fluctuations due to heat stress. For example, PI, phosphoinositides, are the only group which had a significant decrease in abundance. As you can see, the PSA plot for the lipids is quite different, with no clear distinction between the two groups. However, it does show that the control group is constant and there is an increased variation in the heat stress group. This is possibly reflective of stress and regulation occurring in the fish. This is also seen in the PLS delay graph, as the R2 value has a more moderate degree of correlation and accuracy compared to the aqueous metabolites. The volcano plot also displays our significantly increased and decreased lipids, again being highlighted in green. The enriched pathway analysis highlights 4 main mechanisms, including phospholipid biosynthesis and steroid biosynthesis in the organic phase. I will now discuss my main findings. The first pathway which was recognized was an increase in catecholamine synthesis. We observed an increase in L-DOPA but a decrease in epinephrine. This was not anticipated, as we expected the epinephrine to rise. However, DOPA is a precursor to epinephrine production. This possibly indicates a stimulation of the pathway. Epinephrine levels vary based on the stimuli, degradation, and storage in the adrenal glands or tissues. Possibly, our stress levels weren't intense enough to stimulate a high epinephrine response, as we only studied mild heat stress. Additionally, DOPA is seen as a biomarker to reactive oxygen species formation, which could lead to possible contributions to oxidative stress as well. We also saw increased cortisol levels as hypothesized. Cortisol has been seen as a primary glucocorticoid released in rainbow trout in times of stress. GABA was also seen to decrease in abundance. This indicates possible regulation between cortisol and GABA, as GABA inhibits cortisol release. Additionally, cortisol has been seen to increase and induce alternative energy models, including gluconeogenesis, which can cause hyperglycemia. In coho salmon that underwent heat stress, they displayed increased levels of plasma glucose and cortisol concentrations 2.5 hours post-stress. Additionally, the 2-oxothreonine degradation pathway was the most enriched. This pathway produces keto acids needed for gluconeogenesis and acetyl-CoA production. This figure shows us amino acids and ketogenic amino acids which can be fed into this pathway. Finally, we noticed higher overall lipid content and cholesterol in the organic phase during heat stress, as seen in previous studies. Cortisol stimulates lipid oxidation, possibly causing an increase in lipid circulation in the blood. Cholesterol is also important in maintaining membrane fluidity, as increased temperatures can cause the membrane to become disordered. Phosphotolins were also involved in cholesterol homeostasis, which were seen to increase in our results, and phosphoinositols are involved in signaling pathways, which can possibly indicate decrease in signaling cascades due to stress. To summarize, we observed an increase in plasma lipid levels, stimulation of stress hormones, and increase in alternative energy production. Possible limitations can include our short observation period of only 1 week of heat stress, as chronic heat stress could have shown more stable results and can be worked on in the future. We can also include threonine as a possible amino acid biomarker of gluconeogenesis and heat stress. I'd like to thank Denise Simmons for allowing me to work in her lab and having an amazing group of lab mates, as well as my parents for providing me this education. Thank you. Any questions?